Είναι αλλού για αλλού. Είναι αλλού για αλλού. Να την Γεωργία.
Ναι.
Γεια σου Γεωργία.
Καλημέρα. Γεια σας.
Είσαι καλά?
Καλά είμαι, εσείς?
Καλά, καλά βρε Γεωργία. Ποιους έχουμε εδώ πέρα? Έχουμε πολλούς που...
Έχουμε τη μεγάλη ομάδα και τον κύριο Πέτερς.
Okay.
Κάναμε ένα τσεκ με την τεχνολογία.
Good morning.
Good morning. Good morning.
How are you? Sorry for my Greek early in the morning for you.
Oh, no problem. I like foreign languages, although I don't speak it at all. I got Kalimera at least. Yeah.
Good, good. All right, we are ready, I suppose, from your side. And
yes, we are.
Okay, and I will be...
We are checking at the same time the social media technical features. We are almost done there as well.
Excellent.
And I have, we will have the same, let's say, flow. I will start briefly, and then I will ask you to give us a short intro in order to introduce Dr. Peters. We check that the presentation is working properly, and we will have, let's say, 40 minutes as we assume presentation. And then if we have the Q&A, if we have questions, you, me, we can address the questions to Dr. Peters in order to reply. We have more than 50, 60 registrations so far.
Okay, good.
Okay. Good, good, excellent. So I put this as the background. Is it better?
Yes, much better.
So we wait like 5-6 more minutes and we are back.
Okay.
Yes, and we will. I will give, let's say, the go ahead in order for the tech team to open. I will give you, the green light, and I will say when we start. So yes, let's be back in, let's say, 6 minutes, and we can finalize the technical issues and start.
Okay, okay, good.
Thank you.
Thanks a lot, Georgia.
Θέλετε να ξεκινήσω το webinar και να αρχίζουν να μπαίνουν οι participants?
Όχι, όχι, δώστε μου λίγο ακόμα.
Okay.
Εντάξει, γιατί δεν θα είναι και όλοι μέσα, οπότε τι νόημα έχει να πέφτουν, καθόμαστε απλημένο. Και 58 το κάνουμε. Έχω το track της ώρας, το έχω.
Ωραία.
Έλα, Μαρία. Δεν πειράζει, ναι.
Okay, I think we have less than 1 minute. Good luck. I will ask the tech team to admit the participants. So thank you very much. Good luck. We can start admitting the participants.
Okay, I start now.
Thank you. Good morning. As we are now admitting our attendees, let me welcome everyone who is joining and watching us. We are back at the webinar series of the fruit flies in silico prevention management project under the Horizon 2020 European funding for research and innovation. As we are progressing to the implementation of our project, we want to share and communicate our results and outputs. So today we will focus on nematodes for off-season control of the Mediterranean fruit fly. One or two technical details about today's webinar. Please note that our webinar is being recorded, as we want to have it for dissemination and communication purposes of our project. And please be aware that after the presentation, we will have some time for your questions. So please leave any questions using the Q&A button that you will find at the bottom of your screen, and we will share them to our presenter. I don't want to lose more time. I would like us to start. I will ask our moderator and project coordinator, Professor Papadopoulos, of the Department of Agriculture, Crop Production and Rural Environment of University of Thessaly, to briefly introduce us to today's topic, and to our speaker, Dr. Arne Peters. Thank you, Professor.
Thank you, Georgia, for the nice introduction. Welcome, everybody. Good morning. I'm really glad today to introduce Dr. Arne Peters, who is leading the research and development team at the INEMA. INEMA is a company specialized in entomopathogenic nematodes and biological control. So Arne has a long history in entomopathogenic nematodes. He started in 1990s and was a founder, one of the founders of the company INEMA in 1997. So besides all the supervising activities, is leading the process development team. He has collaborated with other scientists. One of them is Apostolos Kapranas. He was formerly in the Biennaki Phytopathological Institute on addressing and controlling other pests such as the large pine weevil, the Hylobius abietis, of course with nematodes. So we are happy to have Arne and INEMA on board on the FF-IPM program. Is really a challenging effort to develop off-season biological control for the Mediterranean fruit fly and other invasive fruit flies as well. So this off-season approach in managing fruit flies is a central concept in our project. And as a coordinator and all the partners, I think we are really happy to see that we have developed tools that can support this concept. So I'm looking forward, as all of the attendees, to the talk of Arne. So Arne, the floor is yours.
Well, thank you, Nicolas, for this introduction to my company. I will just share the screen now and start my presentation. I think you can all see the presentation now. So I will start with my topic, nematodes for off-season control of the Mediterranean fruit fly. And I will actually, because I'm not sure how familiar everyone in the audience is with nematodes, give an introduction to entomopathogenic nematodes first. Also tell something about the mass production, how does that work, and then I will spend some time on ways to apply nematodes for insect control, because I believe that there is a lot of room to widen the nematode use by suitable and clever application methods. And then I will come to the trials to control Mediterranean fruit flies, which were carried out during the time of this project in the lab and in the field, and conclude with the current recommendations for using nematodes for the off-season control of Mediterranean fruit flies. Okay, so what are nematodes? Well, you're probably aware, at least if you're a zoologist, biologist, that nematodes is a very important class in the animal kingdom. It's supposed to be the richest in terms of individuals from all the animals. And there are a lot of free-living forms mainly, but also quite a few parasitic forms there, which either are parasites of plants, of humans, of livestock, of pets as well, and of insects. I mean, that's where we come in. There are insect parasitic nematodes. The most well-known are the nematodes. Those are nematodes which are really parasites in the classical sense that they do not kill the host until very late. So they are not ideal candidates for biocontrol. But the nematodes we are working with, the so-called entomopathogenic nematodes, so pathogenic rather than parasitic, they are killing the insect within a quite short time. And I will come to the details now why they are able to do so. So the entomopathogenic nematodes, they are symbiotically associated with entomopathogenic bacteria. So basically, the nematodes are bacteria feeders, and there are a wide range of bacteria feeders among the nematode species. And it's easily conceivable that they have, at some point in the evolution, encountered an insect pathogenic bacteria, which has given them an enormous advantage. And as the evolution was going on, they developed a quite close association with these entomopathogenic bacteria. They kill the insects within 24 to 48 hours post-infection. And inside the insect, they actually feed on bacteria rather than on insect tissue. So here you can see a picture of the bacteria inside an infective juvenile. They are, in some species, they are actually enclosed in such a pouch here in the intestine. Otherwise, the infective juvenile is not feeding. They are living in the soil and waiting for insect hosts and also actively seeking the insect host. And once they have reached them, they will actually release these symbiotic bacteria. So I go to the life cycle. It's a rather simple life cycle. Basically, the infective juveniles, they are in the soil and they enter the insect host. Then they release the symbiotic bacteria and develop to adults. The insect dies and the nematodes proliferate, and they feed on the bacteria which are actually digesting the insect tissue. And after a while, depending on the size of the insect, it's one generation or more generations of nematode cycles. The infective juveniles emerge from the host cadaver, from the depleted host cadaver, and then they are ready to find and infest new insect hosts. So if you look at the single step of this life cycle, the most interesting in terms of biocontrol is obviously how do the nematodes encounter the insects, and they have different ways of doing so. So, for instance, this Deinonyma garpocapsae, especially, one of the species that has its habit to stand on its tail and to protrude its body, and by doing so, it's waiting for insects passing by. And if an insect passes by here, it sticks to the insect and can then look for the entry points. So, but they also move through the soil, and there are a number of cues which they use to find the insect. Noise has been identified, so the noise the insect is doing while it's chewing on the roots, the movement of the insect itself, so the mechanical vibrations. Then there are chemical cues from the insect or from the injured roots the insect is feeding on, and then carbon dioxide. This is all for insect larvae in the soil. Obviously, for insect larvae on the leaves, that's a bit different. But for Mediterranean fruit flies who are, for instance, feeding inside a fruit, some of these cues are also important cues for the nematodes to locate and find the insect larvae. The nematodes can move in galleries if the galleries are small enough, and they can also move inside the fruits, which is very important because the larva has obviously left a gallery which the nematode can follow to find the larva. Once the nematode has reached the insect, it can penetrate inside the body, and it actually must reach the insect's hemocoel before it can release the bacteria. There's no sense of releasing bacteria inside the intestine of the insect, for instance. And well, they do enter through natural openings like the mouth, the anus, or the spiracles, but they can also enter directly through the skin if the skin is not too thick and if the skin is not covered with a wax layer. So that's also happening. I have a short movie which I will probably play accelerated now, which shows the infection of different larvae, a beetle larva, the western corn rootworm. There we have actually made a movie of how the infection is going on. So I will comment while it's running. I hope you can see it. You just saw the larva on the ground, moving and waving. And these nematode larvae, they infect the insects. And they have actually already entered here the insect, which has died. And you can see, if you look very closely, the nematodes moving inside the insect. Here you can see it better. So the insect is already dead. The bacteria have been released. You can see the nematodes moving inside the dead insect. I think there's another picture where such an, here where such an insect has been squeezed under the microscope. And then, yeah, the nematodes obviously are squeezed out. You can see that these nematodes have already started feeding. Well, you can probably not see it, but I can see it because they look a bit different from infective juveniles. And if the time proceeds, these nematodes grow, and the insect carcass at the end is filled with nematodes. And also with some of the species, color is produced. Those are the symbiotic bacteria here producing a red color. So it's easily recognizable if a nematode is infecting some insects by the red color. Yes, and after a while you see the very big adults inside the insect. And these adults carry, well, hundreds of eggs inside. Here you can see these small round bubbles. Those are the eggs. Nematodes can release the insect at this point. Usually they stay inside the insect until the infective juveniles have been developed. So I will jump through this and show you one of these females with all the eggs inside. So it's filled with the nematode eggs. And as I said, in one generation, we have a multiplication of more than 100-fold. And the proliferation and the growth of these eggs and the hatching of the eggs is so quick that the mother actually has no time to release all the eggs. So the nematodes start feeding inside the mother body. So basically it's also consuming the tissue of the mother. And at the end, all the infective juveniles are released from the mother insect. So those are already infective juveniles. They are very dark because they are full of lipid, which serves as an energy source for the, well, unknown time which they will need in the soil to find an insect. They can survive in the soil with these lipid reserves for 6 months, depending on the soil temperature, also 1 year. And they while they are in the soil, they do not feed, so they cannot take up any nourishment inside the soil. And here is one of the Diabrotica larvae filled with all these infective juveniles. Out of such an insect, we get about 100,000 infective juveniles hatching, and it's enough if 2 or 3 nematodes infect these insects. So through multiple generations, you can have an enormous reproduction. Okay, I hope you enjoyed this video, although it's lunchtime. I will now shortly show what do we do in mass production. We could, for one, produce them in insects, and that's actually still done in some regions of the world, since the insects they reproduce very well in insects. But obviously, insect rearing itself is an expensive and labor-intensive task. So we do it in vitro in liquid culture. And to do so, we pick nematodes from the soil samples. This is infective juveniles with the bacteria inside. And these infective juveniles are then infested to insects, and the bacteria are released. The insect in the beginning is still alive, which is good because then the active immune response of the insect is actually removing all the other bacteria which inevitably come into the insect during the penetration process. So we can quite easily pick the symbiotic bacteria from a freshly infected insect and isolate it on an agar plate. It's more difficult to get sterile nematodes because we have to wait there until the insect is dead and until the nematode has actually developed eggs. And these eggs need to be surface sterilized because at this point of time, the insect is already quite decomposed and the intestine tissue is open. So there are a lot of other bacteria in the carcass. So we do get out the eggs from the females, and then we incubate the eggs in nutrient broth for a while just to check. Well, no, first of all, we surface sterilize the eggs, and then we put them into nutrient broth to check whether the eggs are really sterile. And then if the eggs are sterile and the young nematodes have hatched, we quickly have to combine the nematodes and the bacteria. The nematodes without the bacteria will not survive for more than a few days. So we have to immediately combine the bacteria with the with the bacteria, so the nematodes with the bacteria, and then we can go on. So this monoxenic culture on the agar plate is basically our starter to run all the mass production. So from this agar plate, we can go into shaken flask, it's standard microbiology, and then into vessels, which are 10 liter, 150 liter, 3000 liter. This is a rather old slide. We now have actually tanks which are 100,000 liter, and we can produce nematodes in that volume. And it's important to notice that all this here is actually a sterile environment. So in all these tanks, we have only the nematode and its symbiotic bacteria, which is very crucial for the success of the mass culture. So how do we actually do the mass production? Is we have a pre-culture of the bacteria, which is a bit different from what is happening inside the insect. As I said, in the insect, the nematode goes inside and then starts to develop and releases the bacteria. Well, this initial signal for development is not easy to copy or to create artificially, because you would need some insect blood. It's very difficult to have this food signal for development. But luckily, there's a second food signal, which is produced by the bacteria culture. So we do a pre-culture of the bacteria, of the symbiotic bacteria, for 1 to 2 days. And then we add the monoxenic nematodes to this culture. And these bacteria produce a signal which makes the nematode enter the development. Those nematodes start feeding here, and they start development to adults. So in this first step is very important. We spend quite a lot of time to optimize this onset of the development of the nematodes by steering the bacteria pre-culture. So once the nematodes have developed, they will then either produce males and females in Steinernema, and these males and females have to mate, and then they will produce eggs. In Heteroptetus, in the first generation, we only have female phenotypes, but these females are also producing sperms. Those are automectic females, which is very important for being successful in liquid culture. So I will show in the next slide. So we have males and females in Steinernema and hermaphrodites here in the genus Heteroptetus. And those, by the way, are the only 2 genera which are produced nowadays as biocontrol agents. So why is it important that we have automatic females for Heteroptetus? They have in the second generation also males and females, but the way they mate is so delicate that it does not work in liquid culture. You can see it here. The male only adheres to the female here at the end where the female, the vagina of the female is. And this is not possible in liquid culture. The male has not a chance to look for this place. While in Steinernema, the male is coiling around the female, and then it has all the time to find the proper space. And this is also possible in liquid culture. That was a very important finding for us, because other companies have started with producing Steinernema. So they thought, well, it's no problem for nematodes to mate in liquid culture. But we started with Heteroptetus, and then we saw there are some problems with working with males and females. So we need to have the automectic females which produce their own sperms in liquid culture. Okay, with all this introduction, I will come to the advantages and limits of entomopathogenic nematodes. They have a very wide host range. Actually, insects are infested, but and they have also a reasonably high efficacy. They have the possibility to move in the soil and to search for insects. It's limited, as we will see later, but they can actually move, which is quite unique in biocontrol agents. And they can be produced in vitro, which is due to their the symbiosis with the bacteria. And they can also be applied with existing application technology. So with normal sprayers, because the infective juveniles are very small, they are only about 0.5 millimeter long, and they will pass through standard agricultural nozzles. The drawbacks of this agent is that they have a very short shelf life, and they need moist conditions. And as a result, they are actually quite expensive because they cannot be stocked or stored for a long time. And also the logistics are a bit complicated because of the moist conditions and the cooling needed during transport. Okay, what are the main nematode markets today? Just very quickly. One very important market is scarlet flies. The larvae are infested by nematodes. The larvae do cause problems in seedling and cutting cultures and in mushrooms. Mushroom is a very important market for us because these insects are a big problem there, and they can be nicely controlled with nematodes. And obviously, the use of nematodes have become very important because non-chemical solutions have been fostered by the consumer's requirement. But in some cases, there are also no chemical alternatives available anymore. As with this insect, the black vine weevil, that's a soil-living insect, at least the larva is living in the soil, and there's no way to control it chemically right now. So you can either dip your seedlings in a solution with nematodes before you plant them out, or you can spray them. You can also apply the nematodes with the drip irrigation and then control here the nematode in rhododendron or in soft fruit here, strawberries. Another very important market are scarabid larvae, which are living in grassland mainly, can also be nicely controlled with nematodes. In the forest, Nicolas was already mentioning this. There's this pest here, the Lucius abiatus, large pine weevil, can also be controlled with nematodes. It's a bit cumbersome to do so because you have to get a lot of water into the forest and apply it directly to the stems. But it's being done in Scotland and also in parts of Northern England and Ireland. Then we come to orchards, which are more close to the Mediterranean fruit fly. Here we can either control the flat-headed root borer, Capnodes tenebriones, in the soil, but here also you have to find ways of injecting the nematodes to the soil, and then once it's near the roots, it will find the galleries of the Capnodes larvae and then follow them and kill the larvae. The codling moth, in a way, is actually very similar to the Mediterranean fruit fly because in the winter it also spends a long time in the larval stage, which is always good for a nematode treatment. So we do apply nematodes against the codling moth in late autumn or early spring. And then the nematodes are hitting the bark where the codling moths do actually hide, and then they find the galleries and go inside the codling moth and you can slowly kill them. Obviously, you need a nematode here which is active at reasonably cold temperatures. And we have Stananema feld here, which works down to 8 degrees, and that's in many regions where apple is grown absolutely sufficient to control codling moth larvae in the winter. There are a number of other orchard pests which can be applied with or treated with nematodes. Here's a list of them. Not all of them are in Europe. Guava weevil, for instance, that's more in Brazil, the pest. Another important pest, which is well known in Greece, I think, is the palm weevil, Rhizophos ferrugineus. It can be applied or treated with Stanleyma carpocapsa. And here, interestingly, the adult is also quite susceptible. The problem or the challenge here is to get the nematodes where the palm weevil is. So you have to apply the nematodes with quite a lot of water onto the top of the palms. You can also install a line where you can inject nematodes here, and they are then dripped up into the palms. And then they will dribble down and move down in the trunk and find the weevils and the larvae. These are infested larvae because they turn orange. But even in broadacre crops, we have developed methods to treat the insect pests with nematodes. The movie I was showing was about the western corn rootworm in maize. And well, obviously maize is not a very high value crop. So we had to reduce the dosage in order to be compatible with other treatments and to make it valuable to do a biological treatment against this pest. So we have a row application with 200 liters of water with a special device here where the nematodes are actually sown together with the corn, which means that the nematode activity must be maintained over about 2 months. And we have actually genetically improved our isolate to be more persistent. And we now know that this method is working perfectly. The nematodes can survive sufficiently long to later on attack the larvae when they are hatching. Interesting devices are always developed for these nematodes. Not all of them have reached the big market, but one of these devices I want to show here, it's against termites, where you put this actually into the soil, and the producer has actually developed here such monitoring window where you can see whether the termites have entered this device or not, because if they have entered, they will chew on the cardboard, and then here this window will change appearance. And then you know you can apply nematodes, because you know there are termites inside, and you would pour the nematodes here in the center. I'm not sure how much this device is used in the US. In Germany, obviously, there are not many termites. So we have limited possibilities to develop this method further in Europe. What we did develop is a method to treat adult insects. In this case, it's a device to treat adult vine weevils, because they have the habit to hide underneath wooden pieces or stones during the daytime. We simply developed a formulation where we put nematodes in a moist gel where they can survive for a long time, and we put this device here in these grooves of a wooden piece and put that on the soil, and the nematodes will then crawl underneath this piece of wood, and the nematodes have enough time to enter the insects. They can actually enter from the top, which means that they usually crawl underneath the elytra of the insects, where the surface is quite, or the cuticle of the insect is quite soft, and they can then enter to the hemisphere. A very similar device we've developed for an arthropod, which is actually not very susceptible to nematodes, wood lice. But if you apply a sufficiently high dose, you can even kill this insect, and we apply this high dose by attracting the wood lice to bait stations here with some fallen leaves from beech trees, which are infested with a nematode mixture of quite high concentration. So the wood lice go inside this device, feed on the leaves, and the nematodes have enough time to enter the wood lice. They will actually not develop inside the wood lice because it's not a suitable host, but they will kill them. Okay, that was a quick ride through the current status of entomopathogenic nematode science, or application more. They are also very interesting models for fundamental science. Within the FF-IPM project, the objective was to develop a nematode-based control tool for the off-season control of the insects. And one of the activities was developing a mulch formulation, very similar to what I've just shown. It's either a wood mulch or a leaf mulch with a high concentration of nematodes inside, and the idea was maybe when the flies emerge from the soil, they have to move through this mulch layer, and they encounter these nematodes in the mulch layer, and then the nematodes will actually infest the adult flies. So that was one topic, one task which we made. We tried to develop such a mulch formulation. Besides, laboratory tests were done to find the most suitable nematode strain, or commercial strain for mat-fly control, to control the major larvae in the soil substrate, and most important, also in the infested fruits. The nematodes were applied to soil substrate, and then the larvae were added, and the fruits infested were placed on top of the EPA substrate, very much like in the open field. And the efficacy was always assessed by counting the emerging fruit flies after 4 to 6 weeks. And in the end, there was also a field testing done to check the performance of the most suitable nematode species in the established concentration. So let's start with the mulch formulation. I showed that picture initially. The mulch formulation is actually a super adsorbent gel to provide the moisture, and a mixture. There's also sand mixed into this, because in the gel itself, the nematodes cannot move from the gel because the adherence forces are too high. So they have to crawl on small sand grains, and then they can actually lift the top of their body and stand on their tails and do this waving movements. And if a fruit fly passes by here, it would pick up one of these nematodes, and the nematode can then find the spiracles and enter into the fly. So we tested the mulch previously here at INEMA in Germany. Since we cannot work here with fruit flies, we do not have a quarantine station. We were checking the performance with our house insect, which is the mealworm, the lesser mealworm, Tinipira molitor. And we checked different recipes for this mulch formulation. It was containing either silver, so it was always containing water, obviously, but we also tried some thickener, crystalline cellulose here, and we were thinking maybe we can reduce the evaporation a bit by also adding rapeseed oil to this mulch formulation. And actually that was working, so we had best performance of this mulch with all these additives. But it was no difference between 5 minutes incubation of the mealworms in the mulch or 30 minutes incubation. Anyway, since this is not percentage mortality, but dead mealworms out of 40, we were coming very close to 100% mortality. So 40 is about here. So there were just a few insects missing. And that was even in the older mulch formulation could be seen. Interestingly, in the younger mulch formulation, the performance was not quite as good, but even here we had about 50%, 75% efficacy. So we were quite optimistic about this method, and we then sent the mulch formulation to Greece, so it could be tested for the efficacy against adult mat-flies. Yeah, this is the mealworms. You can see 20 means 50%. We always work with 40 mealworms. You can see that there is already after 5 minutes incubation quite a lot of mortality. Interestingly, it does not increase over time. So you do not reach all of them, which, yeah, it's a bit skeptic. So when we tested it on mayflies, the mulch was applied as we had the idea to apply it. It was applied as a layer on the soil substrate, and then the larvae were first put on the soil, and then the larvae actually had to move through the layer 2 times. They had to move down and up again. But unfortunately, there was no difference in the adult flies emerged in control versus mulch. So the efficacy against the mayflies was much lower. However, we did see an effect on the longevity of the emerging mayflies. You can see it here, the survival function. The red curve is the survival of the treated mayflies, and they have survived shorter than the untreated. Well, not enough probably for a field application, but we wanted to know whether with a higher concentration of this mulch, we would have better effects. So we directly immersed the pupa into the mulch and then saw whether the emerging flies would be infected. And here we actually had a significant difference in the adult flies emerged from the control versus the mulch treatment. And again, the emerging flies, which were not dead immediately, like this person, were dying much quicker. So apparently they are actually quite resistant to the nematodes. They can survive with nematodes inside for quite a long time, but they are eventually also killed. But the problem here is we can, in the field, not immerse the pupa directly into the mulch because the nematode dose would be much too high. So we decided within the project to cancel this idea. So we were not working with the mulch formulation in the field because they would not be payable, commercially viable. When we tested the nematodes in the lab, we screened different species, and to make it short, we found that Nematodes is the most infective species in the laboratory. The emerging flies here were much lower when the nematodes were freshly in the substrate, but even if we added the larvae later on, there was a residual infectivity of the nematodes in the substrate for 2 and 4 weeks. So in conclusion, the cumulative mortality of the mayflies was 50% at 20 degrees, and we have a residual activity of the nematodes in the substrate for 4 weeks. And all the nematode species significantly reduce the larvae inside the fruits as well, which was a very important finding because the larvae seem to stay in the larval stage in the winter in the fruits for a long time. Whereas in the summer, the larvae immediately turn to the pupa. The pupa are not susceptible to nematodes. The surface of the pupa provides no entry points for the nematodes, and it's too hard for them to be penetrated directly. So it's a very big advantage that the mayflies has this habit to live as a larva in the larval stage in the fruits during the winter. No big difference between the species here in the infection of larvae inside the fruits. In terms of temperature, Asphaltia is the most suitable for the winter time because those two species, they have troubles to infect insects at temperatures below 15 degrees. There's another species which can actually infect below 15 degrees, which is Heterodytes downsy. So we also produced this especially for this project and had it checked, but the performance of Heteroclitus downsy was not better than Steinema feltier. So we were sticking to Steinema feltier for the field trials. And the final dose we came up with was 150 and 250 nematodes per square centimeter. And that dose was then transferred to the field, basically. Yeah, that was the most successful at 15 and at 25 degrees. So in the treatment early season is spring season. Two orange types were treated, Neville oranges and sour oranges, in March, end of March. And the dose was, as stated before, here expressed in IJs per square meter, 1.5 million or 2.5 million infective juveniles per square meter, and half a liter per square meter. The fallen fruits were equally distributed between treated and untreated parts of one tree. And so there were a total of about 50 trees and 49 trees treated. And for every tree, half of the area was left untreated and half of the area was treated. And then after the treatment, cages were erected and flies were counting 3 times over a period of 7 weeks to see whether there were differences in the number of emerging flies. Those are the results of this field trial. It was differentiated by the tree type. The sour oranges gave these results. The cumulative number of adult medflies emerging was higher in the control than in the nematode treatment, significantly higher actually in all cases, not just in the sour oranges, but also in the case when the Neville oranges were taken together with the others. We had a significant reduction of the emerging fruit flies. There was no significant difference actually between the 2 doses, which is good because we can save quite a lot of money by moving to the lower dosage. So the 1.5 million per square meter is a sufficient dose to control the medflies. Another trial was done in autumn in Corinthos on Valencia oranges, so just one type of oranges in this case, in October. So that would be also off season, but in autumn. With in this case just the higher dose. For some reason the lower dose was not tried in this trial. It was probably because it was the first trial done. The fear was that we would not be successful, so we did not include the smaller dose here. Again, the fallen fruits were equally distributed between treated and untreated part, and 50 trees were treated. Each tree was divided in 2 sections, treated and untreated. The cages were erected a few days after treating the areas, and the flight counted during emergence. Here a bit, not quite as long as in the previous experiment, simply because the flies do not emerge in winter. They need a certain temperature. So until 22nd of November, the flies were counted. And again, there was a significant reduction of emerging flies by the nematode treatment. Also, definitely above 50%. So in conclusion, the off-season treatment with nematodes does work. It's a suitable tool to control medflies off-season. We get approximately 60% reduction in fly emergence in the lab and in the field, which was very relieving that the field trials match even in terms of the magnitude of the control, the lab trials. The application in spring or autumn is favorable because we have usually higher soil moisture there, and the temperature is more suitable for Nematodes here. And well, there are other reasons why it's favorable, because the larvae spend a longer time as in the larval stage. They are not developing into pupa so quickly. If you look in the lab, in the published papers, you will find a lot of papers on nematodes used against Mediterranean fruit flies. But most of them, actually, they consider local nematode isolates and not commercially available isolates, which is a pity, but it's understandable, because usually this work is done by people working on entomopathogenic nematodes, and it's quite easy to isolate your own isolates. And then, I mean, the scientists want to see what are these isolates capable of. It's a pity, because to get these isolates into mass production is not an easy task. And since mass production is done by really big companies, they are not interested in a very scattered range of different isolates to be produced. To give you an idea, all over the world, there are now, I think, less than 5, well, maybe 6 species of nematodes produced. And the isolates within each species, there are often only one, because the companies, they copy from each other, or they have just seen this particular isolate is most suited for mass production, while having also the best infectivity against a wide range of insects. So it was it was our intention to work with commercial isolates here in this project, to be able to recommend at the end a method with nematodes which are available commercially. Very important is that the larvae inside the fruits are infected. The pupal stage is not infected, and we had this when we were working with cherry fruit flies in Germany. We've seen that the pupation is taking place so quickly that the nematodes hardly have a chance to control the larvae. When the larvae come out of the fallen fruits, they immediately come out after the fruit has fallen, and then it's just a few hours before the pupal stage is formed, and then the nematodes have no chance infecting. But the medfly, especially in the winter, has a long time in the larva stage inside the fruits, and that's a good entry point, or good area for nematode treatment. The project really showed that the nematode treatment is a very robust method with similar results in spring and autumn. There will be another trial evaluated, I think, soon. It has been done in spring in Greece. The effect on the summer population of such an off-season control with nematodes is obviously dependent on an area-wide treatment of infested hotspots in an orchard area. So it requires a lot of knowledge about the hotspots which need to be treated to stop the buildup of populations. With that, I think I'm done, and I want to thank you with this nice picture, not from an orchard, but from the area where I come from. We grow a lot of oilseed rape, and you see here some parasitic wasps, which are after the pollen beetle in the flowers. Thank you.
Thank you very much. I'm checking the Q&A if we have something from our participants. I don't see anything for now. If you want to share your thoughts or any questions, now is the time, please.
So Arne, thank you very much for this nice presentation and the introduction also to the industrial part of producing the nematodes, which is quite interesting and challenging. I have actually a question. So how you think like a company of the future of the management with entomopathogenic nematodes, with all your developments? You see like this is going to increase, or what are the challenges that you will face, considering also the European policy for reduction of pesticide use and for achieving these goals of the Green Deal?
Yeah, well, I think there's a great future for using nematodes. Although I must say that we were a bit blinded by the past, because we were mostly applying nematodes in niche markets, in high value crops. And there we did not really encounter the chemical industry so much as we do now, while we are entering broad acre markets like corn, for instance. We feel a strong resistance, let's say, from the agriculture and also even from the government, which is probably steered by the chemical industry, to adopt the method, despite the European guidelines to replace chemical methods if biological methods are available. It's happening very, very slowly. And this is especially true for well, commercially interesting crops, or for crops which are commercially interesting for the large companies. That's something which we underestimated, or I at least underestimated. So, but the whole business of biocontrol is growing, and so we are also starting to organize ourselves and make our own lobby work.
Okay, I've seen actually some questions coming in. So
yeah.
The first is that from Sajjad Mir, that it's very nice presentation. I appreciate the presented for his comprehensive work. Is it possible to get 60% reduction in laboratory and field conditions? Arne, this is the question.
Yeah, it was possible in these trials. I mean, definitely this work was very carefully done, and the doses were directly transferred from the lab to the field. And usually what happens is that you get a lower efficacy in the field, because obviously the soil layer in the field is much higher, it's much deeper as in the lab trials. But in these trials, we got exactly 60%, and that was almost exactly what we also got in the lab. It will be difficult to get any higher, which is evident by the fact that we did not see any dose effect. We did not see any difference between the almost double dose of 2.5 million per square meter compared to the 1.5 million. So 60% seems to be the limit. We are not going to reach 90 or 100%.
Yeah, but there is another follow-up question actually regarding whether pupae of tephritid fruit flies are infected by nematodes. I think you have commented already on this.
Yeah, unfortunately not. The pupae, although in some papers you find the statement that the pupae are infected, what I believe is what happened there is that probably some of the pupae were not really in the pupa stage yet. The pupae are not infected, but you can obviously find pupae with nematodes inside, because if they are infected in the very, very late larva stage, they will be alive, they will produce pupae. And what you then find is pupae with filled with nematodes, and the nematodes have a lot of troubles to get out of the pupa again because the skin of the pupa is so dense.
So also there is a question of Kostas Zarpas from our group here in the University of Thessaly. Kostas mentioned that we have applied the 2.5 million nematodes and whether the 1.5 million is sufficient for off-season dose. So again, you have commented, but we would like to have your opinion on this.
Well, the 1.5 million, there was no difference to the 2.5 million in the one trial where these two doses were compared. So from these results, 1.5 million is by far sufficient. We don't know whether we see a dose effect or at which concentration you would see a dose effect, but between these two, we didn't see. Maybe we can even go lower, but 1.5 million is the recommendation now.
This actually triggers me to ask about the cost of the operation, because it seems that nematodes are quite costly. And do you think that in the future the prices can become lower, so can be adopted more widely by growers?
Yeah, the nematodes are costly. That's right. And to be honest, in most applications, we actually apply an even lower dose of 500 million per square meter to be able to, for the grower to pay the nematodes. And maybe we can go even lower for the fruit fly. So that's one way of reducing the cost for nematodes. Another thing which is happening is with the increasing size of the production, the prices for nematodes are continuously dropping as well. Yeah.
Good. And I've seen another question from Apostolos Kapranas, who was part of the development.
I thought very good. Yeah. Hello, Apostolos.
Apostolos says, hello, Arne.
Well, he's done most of the work, so my regards and my sincere thanks to the very good and precise work of Apostolos on the lab trials and also in the field trials.
Apostolos is asking if there is a very effective strain found in Mediterranean that acts on much lower dose and there is market demand, then you think it would be still impossible to bring this to mass production. So that's
okay. Well, no, not really. It's not impossible. If the market is sufficiently big, you will find a company who does the production. We recently started the production of this Heteroptetus downsy because we thought we need something for the winter season and against vine weevils. And that's still a very small market, but we did start the production. And it's absolutely thinkable if the advantages of another isolate are very, very pronounced, that and if the market is sufficiently high that the production is taken up.
Good. Yeah. And I've seen actually probably the last question here is which juvenile stage of nematode is most effective against which instar of the maggot. So we have clarified that the nematodes are not effective on the pupae, but they are on the maggot. So that's
they are on the effective on the maggot, and it's actually an important question because the maggot size is also affecting the infectivity of the nematodes. The late instars of the maggots seem to be more susceptible to nematodes, which is, it's not like this with all insects. It seems to be a matter of the size. So I've seen that with other insects, very, very small larvae are less susceptible than larger larvae. In terms of the juvenile stage of the nematode, there's only one stage which is infective. That's the dour juvenile stage, or we call it infective juvenile stage. It's a specialized stage of the nematode which does not take up any nourishment, lives on lipid reserves, but has this behavior of looking for insects and penetrating into insects. And it's the only free living stage which is available, and it's only this stage which we can formulate and sell. Yeah, others cannot be sold.
Good, good. Thank you very much. There is also a comment of Mohan Kumar. It's like also awesome presentation and nice work by the doctor. So that's the final comment I think we have.
Thank you.
So with this, before Georgia's last words, Arne, I would like to thank you very much. I think we have learned a lot today, and we have seen that with all this development, we can definitely have an additional tool to target medfly off-season and probably other fruit flies. And apparently this is a component of a system, and we are really glad that this has been developed. And also I've seen that the future of nematodes with all the new formulations, the entomopathogenic nematodes, is quite good. So thank you very much. So something for people attending, it's this is a series of webinars, and we have already planned the next one. So stay tuned. We have more developments of our project as we are heading to the completion of the project. We have much more to report. So thank you very much, Arna.
Yeah, bye. Welcome.
Thank you very much, Dr. Peters. Thank you, of course, Professor Papadopoulos. Thank all of you for joining us today for this insightful webinar. I believe the same as well. We appreciate your active participation through the Q&A and your engagement throughout this session. We hope that the information shared and the discussion held have been valuable to you. As we conclude, as Professor Papadopoulos mentioned, we encourage you to continue exploring for updates via our social media and our webpage related to our project, because we will announce and as we want to have more connection with you through another webinar and, of course, an event that we will announce quite soon. Thank you very much. We will be in touch. Thank you.
Bye-bye. Thank you, Georgia.
Bye.